Bismillahirrahmanirrahim. Today we are going to discuss a very important topic in relation to the cell injury, and the topic for today is the apoptosis, which is also named as programmed cell death when it was first recognized during embryogenesis. There are two types of cell death. One is the necrotic cell death and the other is the apoptic cell death. In the necrotic cell death, when a cell is injured and if the injury is less severe and if the cause of the injury is removed, there is a chance that the cell may return back to its normal anatomical and physiological status. But if the injury is severe, or if the cell is carrying an inherited defect, what will happen? That the cell will die by the necrotic cell death. In the necrotic cell death, what happens? That the cell becomes swollen mainly because of the loss of ATP from mitochondria, which is needed for the ATP dependent membrane transport pumps, and the cells become swollen, and ultimately the cell ruptures the nucleus DNA, and the cytoskeleton and organelles, they are degraded, and in this type of cell death the cellular contents, they leak out into the extracellular spaces, and this type of cell death, which may be caused by many causes including ischemia, results in the release of cellular contents into the extracellular spaces, and this will invite the phagocytes and will induce a state of inflammation, and in this state of inflammation potentially there will be damage to the healthy tissue and cells. The necrotic cell death is always pathological, and it is mostly the result of irreversible injury. Coming to the apoptic cell death, we can say that the cells that are not needed more, and those cells, they are damaged beyond repair. What happens here? That the cell activate enzymes, and these enzymes within the cell, they will cause degradation of the DNA, degradation of the nuclear and cytoplasmic proteins, the cytoskeleton of the cell, and as a result the cell dies in a more neat and clear way without inducing inflammation and causing injury to the surrounding healthy cells and healthy tissues. Here what happens? That the cell which form blebs, and these blebs are broken down away from the cell, known as the apoptic bodies. Now these apoptic bodies, they are enclosed in membranes, and there is no chance or very little chance of the leakage of the contents, intracellular contents, or the contents of these apoptic bodies, which represent fragments of the cell that has been broken down by the apoptic cell death, and very silently they attract phagocytes, and the phagocytes engulf these apoptic bodies and degrade them, and as a result there is very little leakage of the intracellular contents into the extracellular space, and as a result there is no inflammation and no damage to the surrounding healthy cells and healthy tissues. So we say that the apoptosis is a programmed cell death or cell suicide, and the cell dies in a neat and clear way without inducing inflammation and damage to the surrounding healthy tissue and cells. Necrosis is always pathological, and it is an induced form of death of the cells, and this always occur in abnormal conditions. While apoptic cell death, also known as the programmed cell death, it may occur in two ways. Even physiologically, there may be apoptosis continued in our body and we don't know them. We don't have any problem or signs or any symptoms as compared to that what happens in necrosis. However, there are certain conditions in which, particularly when the cells are exposed to radiations, ultraviolet light, or many other conditions like when the cell DNA is damaged and when it cannot be repaired by the DNA repairing mechanisms, the cell will induce the apoptic cell death and the cell performs a suicide. So, by definition, we can say that apoptosis is a regulated mechanism of cell death that serves to eliminate unwanted and physiologically unwanted and irreparably damaged cells. And there is very little possible host reaction in the form of inflammation or immune reaction. Sometimes apoptosis and necrosis coexist, and the cell death that is initiated by the apoptic cell death may continue later into necrotic cell death. So what happens in apoptosis, that the enzymes are activated and they degrade cellular proteins, structural and enzymes. Uh, they degrade the DNA, and the enzymes that are initiated for apoptic cell death are known as the caspases. The caspases are activated in two steps: the initiation phase and the execution phase. And as a result, these caspases cleave the proteins that make the cell: structural proteins, nuclear proteins, organelles, and they also destroy the proteins that make the cytoskeleton of the cell. By what we mean that the inside of the cell is kept intact and in shape by the cytoskeletal proteins, and when these proteins are broken down, the cell will undergo apoptic cell death. Finally, when an apoptic cell death dies, it is broken into membrane bound fragments known as the apoptic bodies, and these apoptic bodies, they have their membranes such that they activate the nearby phagocytic cells, and these phagocytes, they engulf these apoptic bodies and they are silently removed. Now, in apoptosis, most of the time the students are asked about the definition, the causes, the morphological structure of the morphological presentation of the apoptic cell deaths and certain biochemical features, and that what happens in the cells, and most of the time the mechanism. So by definition, apoptosis is a pathway of cell death which is induced by very tightly regulated intracellular program in which the cells that are destined to die, what they do, that they activate enzymes, caspases, and these caspases in the initiation and after the execution phase, they degrade the cell's own DNA by activating the DNA nuclear endonucleases, the enzymatic proteins, and also the enzymes that degrade the cytoplasmic proteins and the nuclear proteins. Here it is important to note that our cells normally have a potential to undergo apoptic cell death. There is nothing new, but all the mechanisms, they are present within the cell. But in normal conditions, in another mechanism which is known as the anti-apoptic mechanism, it will be, in normal cells, it will be more powerful and it will neutralize the pro-apoptic mechanisms, and the cell remains normal and in a healthy conditions. But in certain situations, when the cell is injured, for example, by sometimes by ischemia, ultraviolet rays, toxins. In this situation, what happens that the pro-apoptic mechanism is activated while the anti-apoptic mechanism is suppressed. Now here the balance will tilt in favor of the apoptosis, and activation of the pro-apoptic mechanisms and suppression of the anti-apoptic mechanisms will result in this type of cell suicide or apoptic cell death. Now what happens in apoptosis, that the apoptic cell death, when it dies, it is broken down into membrane bound apoptic bodies, and the membrane in the apoptic cells and apoptic bodies, it remains intact. By that we mean that the intracellular contents are not released into the outside, but the membranes of the apoptic bodies is altered in such a way that the cell and the fragments, they will activate the phagocytes, and the phagocytes will remove these apoptic bodies by engulfing them and to clear the area. Now we know that what cells will initiate apoptic cell death and what cells are supposed to die by the apoptic cell death. So this can happen physiologically and as well as pathologically. So here we can say that the potentially harmful cells, for example, during the our immune system, some cells, for example, the self-reactive lymphocytes, they should be removed by the apoptic cell death, because if these self-reactive lymphocytes, if they remain in active state, they will induce an autoimmune reaction in our body and will damage the body. So we can say that these are potentially harmful cells and they should be removed by the apoptic cell death. Another example is the cancer cells, which are exposed to apoptic cell death by, for example, cytotoxic T lymphocytes or CD8 cells, and physiologically the cells that have outlived their usefulness. For example, the neutrophils after acute inflammation, and the lymphocytes in chronic inflammation and after the process of healing. So there is no need for these cells further, and they will undergo apoptic cell death to maintain the homeostasis. Sometimes pathologically the cell is damaged in such a way that the DNA repairing mechanisms cannot make a repair of the cells to turn it back to a normal cells. So those cells that are damaged beyond repair, particularly when the damage affects the cell DNA or cellular proteins, these cells will undergo apoptic cell death. What happens that the cells that is destined to undergo apoptic cell death, it will activate enzymes known as the caspases, because they will break the DNA at particular sites, and As a result, when the cell proteins and cytoskeletal structure is degraded, the cell shrinks, and finally the blebs, they are formed in the cell that is undergoing apoptic cell death, and the cell is broken down into the apoptic bodies, and these apoptic bodies in their membranes, they will attract phagocytes, and the area will be cleared when these apoptic bodies are silently engulfed by the phagocytes, and there is no or very little inflammation. Now, what is the function of the caspases? Caspases are the initiation caspases. There are two phases, phase of initiation and phase of execution. These caspases, they cleave the proteins of the nucleus, proteins of the organelles, and proteins of the cytoskeletal structure, and when the during the execution phase, when the endonucleases and other proteins are activated, they will cause the apoptic cell death. Now, there are certain conditions physiologically when the cells are supposed to undergo apoptic cell death, particularly when these cells are not more needed. So here the very important point is the cell destruction during embryogenesis. My dear students, a carpenter and an architect make a rough map first, but later on, by removing the unneeded, not needed things from the map or from a shape, the final outcome is in the form of a very clean, clear, and beautiful things. The same happens during embryogenesis when we get a baby with all its anatomical and physiological uh, status. So many cells that if they are not needed, they undergo apoptic cell death and finally give a good and beautiful functional shape to our organs. The second condition in which apoptic cell death occurs is, for example, those cells they depend on the hormones. For example, the uterine endometrium during the menstrual cycle, we know that the two hormones estrogen and progesterone are required. The estrogen cause development of the uterine endometrium, and the hormone progesterone continue it to maintain the uterine endometrium. But the life of these active endometrial functional cells depend on the supply of the estrogen and progesterones. And uh, in a female, if fertilization doesn't occur, if the female doesn't become pregnant, what happens that the supply of the estrogen and progesterone is reduced, and finally there is no estrogen and progesterones, and these cells they don't get a stimulus to remain alive and functional, and they undergo apoptic cell death and are shed during the menstrual flow. The third example is the proliferating cell populations. We know that regularly our skin cells they become mature, becomes old. The similar is the intestinal epithelial cells or crypt epithelial cells. They mature and becomes old and are replaced by new cells. So these old cells they undergo the apoptic cell death. The fourth example is the cells that have served their usefulness. For example, neutrophils after acute inflammation and lymphocytes after chronic inflammation and healing. When they are not needed more, they are induced the apoptic cell death. The potentially harmful cells, for example, the self-reactive T lymphocytes. We know that in each person, the T lymphocytes can recognize the self and non-self tissue. But if these cells, if they recognize the self tissue as non-self, what that happens during autoimmune reactions, they will cause severe damage, and there are many examples of the autoimmune diseases. For example, rheumatoid arthritis, rheumatic heart disease, lupus erythematosus, systemic SLE, systemic lupus erythematosus, and many diseases in which the immune system recognize the self cells as the non-selves and damage the self tissue. So those cells known as the harmful self-reactive cells, normally they will undergo apoptic cell death to protect the body from the autoimmune damages. Another example is the cell death which is induced by cytotoxic T lymphocytes. Particularly these cytotoxic T lymphocytes, they cause apoptic cell death in viral infected cells and the tumor cells. And if they do not undergo this apoptic cell death which is induced by the cytotoxic T lymphocytes, these cells can be very harmful and damaging to the body. Now, pathologically, what cells are directed to undergo apoptic cell death? Number one is the DNA damage. If there is severe damage to the DNA, or damage beyond repair, when the DNA repairing mechanisms cannot repair the cells, these cells are directed to undergo apoptic cell death. Cells with accumulation of misfolded proteins. There are mechanisms in the form of chaperones and proteolysis that we will discuss later on, in which the misfolded proteins are removed. But if the production of the misfolded proteins which are not properly folded amino acid sequence in a protein, and that is known as the misfolded protein, if a lot amount of misfolded proteins, if they accumulate in the cells, they can cause very severe damage, and many associated diseases are concerned with related with the accumulation of misfolded proteins, and these cells normally they are directed to undergo apoptic cell death. Sometimes the cells that are infected with viruses, for example, we know that viral infection is an intracellular virus, is an intracellular pathogen, and these cells are also directed to undergo apoptic cell death. Sometimes, for example, the glands like pancreas, parotid gland, if there is an obstruction of the duct and there is no outflow of the cellular secretions, so what will happen that these parenchymal cells they become atrophic. We have studied atrophy, hypertrophy, hyperplasia, metaplasia during our previous lectures in which we studied cellular adaptations. But if there is an obstruction and no outflow of the tissue secretions, these cells become atrophic, particularly the glandular organs, and these cells are supposed to undergo apoptic cell death. Now, what happens when a cell dies by the apoptic cell death? We have studied that the cell death involves the cell cytoplasm membranes of the organelles, and the nucleus. So what happens that in apoptic cell death there is condensation of the nuclear chromatins, and we have studied that the nucleus will undergo either pyknosis, karyorrhexis, or karyolysis. And what happens in apoptic cell death that the pyknotic cells which first become shrunken and are broken into pieces, and this process is known as the nucleus fragmentation is known as the karyorrhexis. Now, when the cells is broken down into the apoptic cell bodies, they will invite and activate the phagocytes, and these phagocytes will engulf these apoptic cell bodies and will make the area clear. There are two types of apoptic cell death. In one type known as the intrinsic pathway or mitochondrial pathway, there is nothing needed from the outside, but the inside of the cell there is activation of the enzymes, activation of the caspases, particularly initiating from the mitochondria, and the apoptic pro-apoptic proteins are activated and they will cause release of the cytochrome C from the mitochondria, and the cytochrome C will unite with a protein known as the APOF1, and that will activate the caspase proteins, particularly caspase 9 in the mitochondrial or intrinsic pathway. Similarly, the extrinsic pathway or the death receptor pathway, the second type of apoptic cell death. What happens here that in this type of death, the receptors that are present in the cells that are supposed to undergo apoptic cell death, they known as the death receptors. And the death ligand which is expressed by the cells of the immune system like macrophages and cytotoxic T cells, the death ligand will bind to this death receptor on the cell membrane, and they will activate inside the cell the death domain, and ultimately both the pathways will lead to the activation of the initiator caspases, particularly caspase 9 in intrinsic pathway and caspase 8 in the extrinsic pathway. And the initiator caspases will then activate the executioner caspases, the caspase 3, caspase 6, 7, and these executioner caspases will activate proteins like endonucleases, which will cause fragmentation of the DNA, and other proteins that will cause breakdown of the cytoskeleton. So we know that the enzymes known as the caspases, they operate the mechanism of the apoptic cell death, and this is known as the caspase cascade. Some caspases are initiator caspases, particularly the caspase 9 in intrinsic pathway and caspase 8 in the death receptor or extrinsic pathway, and they will lead to the activation of the executioner caspases, of which caspase 3 is very important, and also caspase 6 and caspase 7. And the executioner caspases will activate enzymes like endonucleases and will cause apoptic cell death. There is another condition which is an inflammatory condition, and which is also carried out by the various caspases. Overall, there are 12 well-known caspase enzymes present in the human cells, and this situation in which the caspases induce an inflammatory pathway is known as the pyroptosis. And this is an inflammatory form of apoptosis, and it is particularly seen in infections with intracellular forms of the pathogens like viruses, rickettsia, chlamydia, and other that will we study in more detail on its own term. Now, what is the mechanism of apoptosis? The intrinsic pathway of apoptosis is initiated when the cell is deprived of the survival signals, when the cell is deprived of the growth factors, if there is excessive DNA damage, or if there is excessive accumulation of misfolded proteins. So all these situations, they activate the various sensor proteins. And these sensor proteins, which are known as the BH3 proteins, they will activate the pro-apoptic proteins, the BAKEs and BAK. Normally, in a normal and healthy cells, when it is not stimulated by these signals, the cell remain in a condition in which the pro-apoptic proteins remain suppressed, and the anti-apoptic proteins, BCL2 and BCLXL, maintain the membrane integrity of the mitochondria, and there is no release of the cytochrome C and other pro-apoptic proteins. But once when a cell is damaged through these mechanisms, the balance tilts in favor of the pro-apoptic proteins, and they are activated, and these bakes and bake proteins, they will make holes in the membrane of the mitochondria. And what will happen, that there will be release of the cytochrome C. We have studied cytochrome C in electron transport system, and we have studied the enzyme cytochrome oxidase, which has very important role in the generation of the ATP in the electron transport system. But here, now the cytochrome C is normally, it remains inside the mitochondria. But when the mitochondrial membrane is damaged by the pro-apoptic proteins like bakes and bake, what happens, that this cytochrome C is released and it comes out of the mitochondria into the cytoplasm. And here it will bind to the APOP1 protein, and this complex will activate caspase 9 in case of intrinsic pathway, and caspase 8 in case of extrinsic pathway. And as a result, the two phases of the apoptosis, the activation initiation phase and the execution phase, will occur, and the cell will undergo apoptic cell death. So in the previous slide, we studied that these types of damages, they will cause activation of the bakes and bake protein, and inactivation of the anti-apoptic proteins. The result will be that the mitochondrial membrane is damaged and it becomes permeable to cytochrome C and also to other proteins that will neutralize these anti-apoptic proteins, and the result will be the activation of the caspases, leading to the apoptic cell death. Now, here we summarize that the BCL2 family sensors, BH3 proteins, they sense the signals, activate the bakes and bake proteins which are pro-apoptic, and inactivate the anti-apoptic proteins, the BCL2 and BCLXL, and as a result, the mitochondria become permeable to cytochrome C and other proteins that will inactivate the anti-apoptic proteins, and the caspases are activated and the cell undergo apoptic cell death. Here in this slide, we can see that the pro-apoptic proteins produce holes in the mitochondrial damage, and the mitochondrial permeability transition pores, they open, and cytochrome C is released, and cytochrome C here combined with the APOP1 protein, and it will activate the apoptic proteins or the apoptic cascade, which will lead to the death of the cell. Now here we briefly discuss the death receptor or the extrinsic pathway. In the intrinsic pathway, nothing was needed from the outside, and the already present pro-apoptic and anti-apoptic proteins, they were imbalanced, and the pro-apoptic proteins were activated and the anti-apoptic proteins were neutralized, and as a result, the caspases were activated, and that will lead to the cell death through the mitochondrial or intrinsic pathway. But what happens in extrinsic pathway, that, for example, here we explain this pathway through examples. For example, the macrophage, it has a lesion known as the tumor necrotic factor alpha. When the macrophage identifies an old cell, pathogenic cells are the cells that have completed their task. Or if a cytotoxic T lymphocyte, if it recognizes in the a virally infected cell or a tumor cells, so the macrophage, it will express a lesion known as the tumor necrotic factor alpha, and the cytotoxic T lymphocyte will express a lesion known as the fast lesion. Now, in the cells that are destined to undergo apoptic cell death, what happens, that they express receptors for the tumor necrotic alpha lesion and for the fast lesion, known as the tumor necrotic factor receptor 1 and the fast receptor, and these receptors are known as the death receptors. Now, when the lesion combines to the receptor, and this complex, which is formed by the combination of the receptor and the lesion, it has an intracellular cytoplasmic region, which is known as the death domain. And the combination of the lesion and the receptor will make changes in the tumor necrotic factor receptor associated dead domain, and known as the FAS associated dead domain. And what happens that when this death receptor domain, when it becomes activated, it will activate caspases, and the cell will pass through the uh, steps of the initiation caspases and the execution caspases, and in this way the death inducing signaling complex that is formed by combination of various proteins, and which will activate caspases, and as a result the cell will undergo apoptic cell death. So this type of cell death is particularly seen in old cells, pathogenic cells, tumor cells, and cells that are virally infected, and we have given the examples of the macrophage and the cytotoxic T lymphocyte that kills these type of cells through the death receptor pathway or through the activation of the death domain inside the cytoplasm, which will be activated after the lesions they bind to their receptors, and the activation of the this protein complex will activate caspases, and the cell will undergo apoptic cell death. Now, as I said, there are two phases. First of all, the cell passes through an initiation phase, and in the intrinsic pathway, as I said, the proapoptic protein, the BAKEs and BAK, they are activated and they damage the mitochondria, and from the mitochondria, by altering the permeability, the mitochondrial membrane becomes open for the cytochrome C, which binds to the FAF1 protein, and it will activate caspase 9. In the death receptor pathway, the death lesion and death receptor, when they do combine, they will activate the cytoplasmic death domain, and ultimately it will lead to the activation of the caspase 8. And these initiator caspases, they will activate the executioner caspases, and the cell pass through the phase of execution, in which particularly the very important is the caspase 3 and also caspase 7, 6 and 7. So these executioner caspases, they will activate nucleases, which will degrade the DNA, and they will activate other proteolytic enzymes that will degrade the nucleoproteins and cytoskeletal proteins, and the cell will undergo apoptic cell death. So in this picture, we can see that deprivation of the growth factors, DNA damage, for example, that is done by radiation, toxins, and the very important free radicals, and accumulation of misfolded proteins, they will activate the BAKEs and BAK, and there will be the release of cytochrome C and also other proteins, pro-apoptic proteins, that will neutralize the anti-apoptic proteins, the BCL2 and BCLXL, and as a result, there will be formation of the initiator caspases. The same we can say that in the second pathway, the death receptor or extrinsic pathway, the tumor necrotic factor receptor, to which the tumor necrotic factor expressed lesion that is expressed by macrophages, and the fast lesion that is expressed by the cytotoxic T lymphocytes, when they unite, they activate the inside dead domain complex, and which will lead to the activation of the initiator caspases. Both the initiation phase is different for the both pathways. Mitochondrial pathway has its own initiating phase, and the death receptor pathway has its own initiation phase. But both the pathways converge at the activation of the executioner caspases. And the executional caspases will activate endonucleases that will cause DNA fragmentation and other proteolytic enzymes that will cause breakdown of the cytoskeleton. And as a result, the cell shrinks and is broken down into the apoptic bodies, and these apoptic bodies, they express lesions for the phagocytic cells, and in this way, when they are attacked by the phagocytes clear the area without inducing inflammation or a severe host reaction. Now, here we can see that in normal cell, when there are proper survival signals, for example, growth factor is present, what happens that the anti-apoptic proteins BCL2 and BCLXL, they are produced and they will neutralize the pro-apoptic effect of the pro-apoptic proteins. And the bakes and bake, they are not activated and they remain in a neutralized state. But here what happens that if there is lack of survival signals, for example, growth signals, cells are deprived of the growth signals, if the cells are exposed to ultraviolet light and radiation, or if there is accumulation of misfolded proteins, or if there is a DNA damage that is beyond repair, what will happen that they will activate sensors in the cell. Here what happens that the BCL2 and BCLX cell, which are anti-apoptic proteins, they are suppressed or antagonized, and the balance tilts in favor of the pro-apoptic proteins. The bakes and bake, which form channels in the mitochondria, there is release of cytochrome C and other proteins that will neutralize the anti-apoptic proteins, and the caspases are activated and the cell will undergo apoptic cell death. Here we can see that in necrosis, the cell membrane ruptures and the intracellular contents are released to the extracellular site, and that activate the inflammation and infiltration of neutrophils, macrophages and other cells, and potentially in necrosis there can be damage to the outside lying healthy cells. But in apoptic cells, all the apoptic bodies that are found by fragmentation of the cell that is destined to undergo apoptic cell death, they are membrane closed. The materials are enclosed in proper intact membranes, and there is no leakage of the intracellular contents, and these membrane bound apoptic bodies will activate the phagocytes and they will engulf these apoptic bodies, and the area will be cleared without inducing any severe host reaction or inflammation. Now, in necrosis, the cell is enlarged, but in apoptosis, the cell will shrink. The size of the cell as well as the nucleus, it will be reduced. The nucleus will become condensed. And we know that the three types of death that occurs in the cells, and particularly in the cell nucleus, is the pycnosis, in which the nuclear chromatids become condensed, and later on there is karyolexis, when the nucleus is broken down into pieces, and karyolysis, in which the nucleus is undergo lysis. But in apoptic cell death, the nucleus will break down into nucleosome-sized fragments. The plasma membrane in necrosis is disrupted while it remains intact in the apoptosis. The necrosis is characterized by the enzymatic digestion of the cells that may leak out of the cell and may induce inflammation. But the apoptic bodies, they remain inside the intact membrane, and the adjacent inflammation is very less or no inflammation, while the same is very frequent in necrosis. Necrosis is purely pathologic, while apoptosis may occur under pathological conditions, as we said, during embryogenesis and during the removal of cells that are not more needed, or those cells that have outlived their usefulness. So apoptosis may occur in physiological as well as pathological conditions. The examples of apoptosis are the growth factor deprivation. For example, as I said, that when the uterus is deprived of the estrogen and progesterone, it will undergo the functional endothelial cells, they will undergo apoptic cell death. Similarly, when the breast cells, when there is no hormonal stimulation, for example, by prolactin and human placental lactogen, they will undergo apoptic cell death and the breast size, it becomes reduced, small and shrunken. And the same is true about the lymphocytes when they are not activated by the growth factors. That is a situation which happens during inflammation and during injury. But when the inflammation is over, these lymphocytes are not needed more and they undergo apoptic cell death. The other example is the damage to the DNA of the cells that commonly occurs during radiation and by the chemotherapeutic agents. We use chemotherapeutic agents during the treatment of cancer, and the main target of treatment is that these chemotherapeutic agents, they induce DNA damage in the cancer cells, and as a result, the DNA damage in the cancer cells lead these cancer cells to undergo apoptic cell death. The accumulation of misfolded proteins in which there is no proper sequence of amino acids, and these misfolded proteins accumulation will occur in various conditions. We will discuss the endoplasmic reticular stress, which is characterized by the accumulation of the misfolded proteins and the damage to the balancing mechanisms. We will study in more detail in our next lecture. But if these misfolded proteins have got accumulated in the cells, these cells are destined to undergo apoptic cell death. And the fourth example is the cytotoxic T lymphocytes. Natural killer cells are CD8 cells. They recognize the cancer cells and virally infected cells, and they release enzymes known as the granzymes. And these granzymes make pores in the membrane of the tumor cells and the virally infected cells, and as a result, these cells are damaged and they undergo the apoptic cell death. Thank you very much.